Perfect. So without further ado, Dr. Laura, take it from here.
Okay, I'll just go and share my screen and start the presentation. All right. So thank you very much, everybody, for attending this presentation. I know that it's evening and you all must be a bit tired, but I'm very happy to see you here. So today I would like to talk to you about this project that I'm working on here at DCU. So just to give you a small background about myself, I am from Italy. I am an ecologist and marine and freshwater biologist. So I moved to Ireland 1 year ago, and I started working on this project, which is called Urban Citizens 632. So what is this project all about? We are basically working together with citizen scientists to gather data about water quality of rivers and streams. And we want to investigate especially places where there is currently a data gap. So we want to see what is the status of our rivers and streams. This project has been built on the experience of previous projects conducted here at DCU, especially Backdrop and the water blitz events. You may have heard about them, or you may have taken part in previous years. All these projects were basically built in collaboration with Freshwater Watch. We are a very diverse kind of team because, as I was saying, I'm an ecologist and biologist, but the project is led by Susan Egerty, who is a geographer, and Fiona Reagan, who is the chemist. So we try to put together our expertise to, in a way, work on this project on different aspects. What we are doing basically is we are asking our citizen scientists to collect data on a monthly basis. So we ask people to, if possible, do a monthly surveys. And these data are being collected for around 18 months. So we started in July last year, and the project will end in January 2025. Many people ask me why it's called 632. So it's basically, this project has been basically inspired by indicator 62. I don't know if you ever heard about the United Nations Sustainable Development Goals. So one of them, the number 6, is called clean water and sanitation for all. And to measure the progress in achieving this goal, we have a series of indicators. So indicator 62 is the indicator that measures the proportion of water bodies with good ambient water quality. So in our project, we want to basically go and investigate this. So how many of our rivers and streams are in good condition, and how many instead need to be basically helped because there are issues, there's pollution, and so on. So this is just to give you the background of the project. We are basically investigating 4 different catchments across Dublin, the Santry and the Torquay in the north, and the Dodder and the Lough Queenstown rivers in the south. All these rivers are in a way discharging water in the Dublin Bay. And for this reason, we chose to investigate how is this water. So what is ending up in the bay and in the biosphere. So just a small kind of information and background about citizen science. I believe that maybe many of you will know what is citizen science, but in case you don't, I just would like to introduce you to this kind of a project. So citizen science is the practice of public participation and collaboration in scientific research. So in a way, through a collaboration between people and researchers or even local authorities, people can contribute to monitoring of natural ecosystems and collection programs. In these ways, we can collect in a way, a lot of data that usually will not be accessible because of funds and resources. And we can, through this data, through this extra knowledge that we gain, we can then go and improve the management of our resources, natural ecosystems, and so on. And the other big advantage of citizen science is that it helps us to detect existing issues. This is true especially in those areas where there's, in a way, data gap. So data are missing and we don't know what's going on. So in the past few years, there have been, let's say, a boom of citizen science projects. I have put here just a few examples, just to give you an idea. So we have citizen science projects a bit around the world, and all of these projects are involving freshwater, but of course there are other projects for many different kind of environmental ecosystems and aspects. So this is to give you an idea of our surveys. What we are asking people to do and measure. Basically, our survey is divided in three big sections. The first is the one that we call visual observations. So we are asking our volunteers to go to a local river of the choice and in a way observe what's going on. We are asking them to collect some ecological information, for example, the land use, the presence of algae, how is the bank vegetation, if there's any signs of pollution, aquatic life, and so on. We also ask to note down some hydrological information because we want to know, in a way, how the weather is influencing the data that we are collecting. And finally, we are also asking for some optical information, especially the water color and the turbidity that can help us to understand also the situation in the rivers. The second part of the survey is instead dedicated to the nutrient test. So we are measuring nitrate and phosphate. And the third part is the macrovertebrates monitoring, which is probably the most, let's say, fun to do, but also challenging because it's not easy to identify all these kind of animals. So I'm going to explain you a bit more details about this nutrient test and the macrovertebrates in the next slide. So what are these nutrients? So we are measuring, in our case, basically nitrate and phosphate, which are essential nutrients for plants and algae. So they are, of course, already present in the environment, in soils and waters. And plants and algae use them to, in a way, grow and perform all the biological processes that they need. There are a few sources that we, as, let's say, humans, introduce to the environment. We basically introduce nutrients through farming, so through fertilizers and animal wastes, through sewage and wastewater, and also in some cases through industrial processes. Why we are measuring these nutrients? Because if we have too much nutrients in water bodies, of course, we can have an issue which is called eutrophication. You may have heard about it. And I'm going to talk about it also later. And just to know, nitrates, if present in drinking water, can have harmful effects. So we should not have them in drinking water. So as I was saying, what is this eutrophication? It's basically a kind of a domino effect that can affect the ecosystems, especially lakes, but also rivers. So what happens is that because when we have, let's say, abundance of these nutrients, so the nutrients level are too high in the environment, all these nutrients will reach water bodies. And what happens is that these nutrients will be utilized by microalgae, so phytoplankton, basically. And these microalgae will start to grow let's say, more rapidly than usual. And they will start to form the so-called algal blooms. So when we have this layer of microalgae floating on the water surface, this will, in a way, prevent the light from reaching the lower level of the water body. And anything which is growing underneath the algal bloom will not be able to receive enough light. So any plant algae which are growing on the bottom will start to die because of this lack of light. And because of this, the oxygen level which are normally present in the water will start to, in a way, be reduced. So once this algal bloom eventually finish its life cycle, all these microalgae will start to basically die and they will sink to the bottom. So once they sink to the bottom, they will start to be basically decomposed by bacteria. So the decomposition process is a process that consumes oxygen. And in this way, even more the oxygen reserves which are in the water will be depleted. Additionally, some of the algal blooms can also produce toxins. And let's say that mostly for lack of oxygen and also sometimes for the presence of these toxins, we can have basically mortality events of fishes and other animals. So that's why eutrophication creates issues. And that's why we are trying to measure the level of nutrients in our water bodies to keep an eye on what's going on. So just to show you an example of what eutrophication can become, I guess you heard last year what happened in Lockerbie and how eutrophication in a way created a big issue there. Until we had, for example, die-off of even big animals. In this case, I just put this picture of this one. We also have fish dying off and so on. So that's why we want to keep an eye on what's going on with these nutrients. How we are measuring nutrients in our project, we are using the freshwater wash kits, which are produced by this company, Japanese company, Kyoritsu, and they are distributed by Wash Europe. We are using basically a sample cup and some testing tubes. What we do very simply is we collect water in the cup and we will transfer this water to the sampling tube. We will leave this tube for 3 minutes or 5 minutes, 3 for space and 5 for space. And after this time has passed by, we will go and see the color of the water. So basically, the more nutrients we have in the water, the darker the color will become. The less nutrients, of course, the paler the color will be. So in this way, we can measure the concentration of nutrients. So I would like to talk to you now instead about macroinvertebrates, which are the third part of our survey. So I don't know if you have heard this very long name, but macroinvertebrates are basically invertebrates. So invertebrates are basically all those animals that don't have a vertebral column, so they don't have a backbone. As you can see from this pie chart, 95% of all non-living animal species are invertebrates, so it's a huge group of animals and creatures. Why we call them macro? Because they are large enough to be seen without using a microscope. So it's easy for us to go and spot them in the field and identify them without a microscope. So macroinvertebrates are present basically in every freshwater ecosystem around the world. They are at the base of the aquatic animal food chain. And what is interesting for us is that there are different levels of tolerance to pollution, especially organic pollution and also habitat degradation. Because of these different levels of tolerance, we use them as basically indicators. And they are the most utilized organism for, let's say, monitoring of rivers. I put here just a slide to give you an idea of the diversity of this community in rivers. We have a lot of juvenile stages of insects which live in riverine waters. We have adult insects, we have different crustaceans, mollusks, different kind of worms, and even water mites, spiders, and sponges and bryozoans. So it's a very diverse community and very rich. So during our project, because it would be not practical to, in a way, teach people about all the different groups of microvertebrates, we have chosen to utilize this method, which is called CSSI. CSSI stands for Citizen Science Stream Index, and is a method that has been developed quite recently here in Ireland by basically the team of Simon Harrison, which is working at University College Cork. Is a method that was developed specifically for citizen science projects and is based on presence or absence of 6 of these macrovertebrate groups. These 6 groups have been chosen because especially they are, let's say, relatively easy to identify, and they are good indicators of either unpolluted or polluted conditions. And of course, we can commonly find them in rivers and streams, so they are not too be that. So who are these 6 groups? We call them the good guys and the bad guys. Not because they are good or bad, but because they can be found basically in good or bad environmental conditions. So the good guys are very sensitive to pollution. So when we find them, it means that basically the water quality in the place, in the area is good. The bad guys, instead, they are very tolerant, so they can live even in polluted conditions. And because of, let's say, less competition and more food available, they can thrive in this kind of conditions. So good guys are the stoneflies, the flattening flies, and the green caddis flies. Bad guys instead are the snails, leeches, and the waterlogs. I put here the scientific name just for you if you are interested to go and see more about these things. So this is just to give you an idea of how the project works and where we are, basically. So we started, as I was saying, last July, recruiting volunteers and starting the training. We have done a few training sessions throughout the first, let's say, 6 months. And once volunteers have been trained, they have started collecting data. We collect all this data, and then we also validate, especially the macrovertebrates data through the pictures that we receive. And we are now in the process of starting analyzing the data. We also try to do workshops and to send, let's say, training material, identification tips, and so on throughout all these months, just to, let's say, consolidate the identification skills, give extra knowledge, and so on. So this is a map of our surveys. We have trained around 65 volunteers, and these volunteers have been collecting data. So roughly 50 volunteers are in a way currently collecting data. To date, we have around 320 surveys from the 4 catchments. I would like to remember that the choice of the sites has been in a way co-created with our citizen scientists, because we believe that they have a lot of knowledge about local rivers, local water bodies, and we wanted to give them the, let's say, the choice of choosing where to go and collect and basically do survey and collect it. Here are some maps from nitrates and phosphates. As you can see, we have a very kind of heterogeneous situation. So some areas have high nitrates and phosphate values. In some areas, for example, these nutrients level are very much varying. So across the months, we have seen big differences. We are just in the process of analyzing this data and start to make sense of what the situation is. But I hope maybe in the next months I can tell you a bit more about our findings. These are a few pictures from instead the macrovertebrates. So our citizen scientists are detecting, of course, the 6 CSSI taxa, and also everything else, so the non-CSSI groups. We are in a way happy, and we are encouraging people to know more about these animals. And in a way, is a good way to build knowledge through their own experience. So whenever they find new, let's say, macrovertebrates, they can always ask us and we can correspond and then go through stuff. So what we are going to do in the next month is basically to, of course, complete the data collection and the data analysis. We want to explore in a way how these variables that we are measuring, so the nutrients, macrovertebrates, individual observations, how are they correlated and what are the patterns, both in sense of time and space. We want to explore potential sources of pollution, and I will talk to you in less time about this. And we also would like to see how this data compare with local authorities' data, for example, EPA, and also with previous projects, for example, our water blitz that was at here in DCU, and the projects from the National Biodiversity Data Center that's also collecting citizen science data. We also would like to evaluate the CSSI because it's quite a relatively new method. So we want to see how it works and what we can do to improve and so on. And of course, we would like to evaluate also our training methods, citizen engagement, and so on, so that we can then improve for the future. As I was saying, we are very interested in this, which are the pollution events. What we have noticed throughout this month is that our citizen scientists have detected quite a lot of these events in basically all the different catchments. So I would like to just show you a few pictures about them. For example, on the River Dodder in Midtown, but our citizen scientists have found a sewage pipe which is overflowing. So they have observed this in November and then throughout the months until January. And speaking with other colleagues and other people, we in a way realized that this issue has been going on for quite some time. So it was spotted also in previous years. In the Century River, we had a few events, actually 4, including oil spill in January, and a series of these, I call them milky suspensions, where the water turns completely milky in February, in April, and even recently in June. Plus, we collected also personal communication from people telling us that there was a kerosene spill and these milky water events also in 2022. In the Kilo de Grange in the south, one of our volunteers have found out die-off for these macrovertebrates. So she went and collected samples, and after 1 month, she came back to the same place. And what she collected was all dead bodies of these macroinvertebrates. And recently, also in the Shangaran River, there was a few events where the water turned basically grayish and bluish in the same spot. Finally, in the talk, we had two fish kill events in May and June of this year. And just a few days ago, the Slange River turned again milky. So this is just to show you how much, in a way, citizens can do in monitoring rivers, because this is all very precious information that would not be noticed if not for people going to the river and observing, taking pictures and reporting. A few of these events have been reported to local authorities, and we have noticed that basically local authorities have, in a way, responded to this and have investigated. So this is telling us that citizen science can be very, very helpful in a way, working together collectively to try to solve these kind of issues. So how all of this can help our rivers in general? The first, as I was saying, is by detecting existing issues and pollution events, so that we know where the issues are and we can then do something to solve them. We have noticed that people are telling us that they feel empowered in this way because they can actively, in a way, do something and contribute to make a change. And of course, is a way to increase knowledge of natural resources and also, of course, awareness of existing issues. So what we would like to highlight is that people can become river stewards. And we have seen things happening, and for example, people continuing with different kind of monitoring initiatives, educating others, speaking to other people, even advocating with authorities and decision makers. So this gives us a lot of hope, and we would like to, in a way, promote this kind of projects because they can be a very good way of helping our environments and giving also opportunity to everybody to take a part in all this. So for today, that's all I have to say. And of course, thanks a lot for listening. And if you have any questions, I'm here. Please go ahead.
Thank you very, very much, Laura. Can everybody hear me?
Yep.
Yes.
Okay, fantastic. So thank you very much, Laura, for that informative talk. So I'm going to pause the recording now. I should actually have pause the recording before I started saying this. So this will be on the internet. Hello. So